Quick question before we get started, Professor.
Go ahead, Matt.
If there was any spelling errors or on the test or whatever, are you going to go manually correct those?
I will manually go over them, and if I can interpret what you're trying to communicate, you will get credit for that.
Okay.
And I was only off by like one letter, and I got the whole thing wrong.
Yeah,
I'm almost not very happy with me.
Yeah. Unfortunately,
That's
that's the
my
nature
problem
of a computer graded system, but I will review everything. Okay.
All right, good.
Okay, good afternoon, everyone, and it's nice to see you. I noticed that many of you have been online at the ClassMarker website and have completed various components of your exam, so that's excellent. As many of you are asking, I do realize there's some stress created when the computer marks a word wrong where it was simply a spelling issue. Please know that I will be reviewing all of those. Okay, so all of those will get reviewed by me, and I hope to have the grades completed sometime tomorrow afternoon. Okay, so when I'm completed with the grading, I will send you an email letting you know that the grades are complete and that you can log back into the ClassMarker website to get your final score. Okay. So certainly bear with me a moment, but I will have those to you hopefully by the end of the day tomorrow. Okay. So do you have any questions right now in terms of what you've experienced or anything else? Okay. If you can hear me okay, please type yes into the chat window. Okay. Okay, great, great. Julia, you have a question. What percentage of the exam is the essay portion worth? It's roughly 50%, Julia. Okay, so I'm just waiting a moment to be sure everybody's online here together. Okay, looks like we do have everybody online together. Fantastic. So for those that are just logging in a little bit late, please know that I will be reviewing your exams and sending you an email sometime tomorrow when the exam review has been completed, okay? So if you haven't taken your exam, obviously you still need to do that by midnight, and I would encourage you, please don't wait to that late hour. Okay, do you have any questions before we start new material? Okay, so for those that have already taken your exam, thank you. And encourage the others to do that early this evening. Okay, so folks, what we're going to do is keep rolling here with an 8-week semester like we have in the summertime. I just keep throwing stuff at you, throwing stuff at you, throwing stuff at you, okay? So please know that we do have lab this afternoon. And next Thursday is your first lab exam, okay? So you can expect that I will send out a lab study guide, if not later today, then certainly by tomorrow at the latest, so that you know what to prepare for your lab exam, which is a week from today. Okay, any questions? Okay, folks, we're going to get into chapter 6, which is the muscular system. So bear with me, I'm going to share my screen with you. Okay, the chat window is up so that you can communicate with me. And the Microsoft Word document is also present. Okay, so chapter 6 is going to be our primary coverage of how muscles function, which is to say muscle physiology. And chapter 6 begins on page 101 of your textbook and spills over for the next several pages. Okay, so you know at this point, okay, so you recall at this point that there are 3 types of muscle tissue in the human body. You had to know that to know a little bit about chapter 4, the tissues that we've covered and which were talked a little bit on today's exam. Those muscle tissues are skeletal muscle, cardiac muscle of the heart, and smooth muscle tissue that you typically find around the gastrointestinal system, you know, around your large and small intestine, around the esophagus, around a lot of organs that control fluid like the stomach, the gallbladder, urinary bladder. These structures all have smooth muscle. Okay, so let's start off by defining common characteristics of all muscle tissue. In other words, it doesn't matter whether it's skeletal, cardiac, or smooth. There are certain common characteristics that all muscle tissue has. Okay. Number 1, all muscles are excitable. Okay, they are excitable. And what that means is, as simply as I can put it, it means that muscles are capable of responding to some sort of stimulation. They're capable of responding to a stimulus. Okay, now that stimulus is usually going to be of an electrical nature, you know, from the nervous system. And it might be of an electrical nature of something that you thought about with your own mind, such as you're telling your arm to move your hand to write some notes at this point, or it might be an unconscious electrical signal where your brain is telling smooth muscle tissue in your digestive system to relax. But muscles are excitable, they respond to stimuli. Number two, muscles are contractile. Okay, that's a fancy way of saying that muscle tissue has the ability to shorten, to get smaller. The third thing is that muscles are extensible. Okay, so we're using some big flowery language here because this is school, this is education, but basically to say that muscles are extensible is a fancy way of saying that muscles have the ability to stretch. Right? I mean, you've probably been in a gym class at one moment in your life where, you know, your gym teacher said, okay, let's do a little bit of stretching before we play football, or do a little stretching before we play volleyball, or do some stretching before we do gymnastics, whatever it might be, right? You know that muscles can stretch. And finally, the fourth common characteristic of all muscle tissue is that, in fact, muscles are elastic. When we say muscles are elastic, think about the elasticity of a rubber band and the fact that you can take a rubber band and you can stretch it out, and then when you release that tension, the rubber band will shorten back up to its normal length. So it can stretch out and shorten back. That's elasticity. Muscles have that characteristic. All muscle tissue has that characteristic. Muscles are elastic. Okay. Muscles can stretch and return to their normal length afterward. Okay. Now, in one of your future labs, not today's lab, but one of your future labs, you're going to get a lot more time learning about some of the muscles of the body, particularly on page 103. So I'm not going to lecture on the picture of page 103, but certainly you should pay attention to it during your study time because we are going to be referring back to it later in the semester, okay? But at least on page 103, you do see some of the major muscles of the body. And as we've talked about before, you know, your book says that there's over 600 skeletal muscles in the body. I think we can make a strong case that there's over 700 skeletal muscles in the body at this point, which is going a long way to say that there are a lot of skeletal muscles in your body, okay? So typically for our anatomy and physiology students that are going into a nursing field or a medical field, we typically require that they memorize the identity and location of about 100 different skeletal muscles, okay? You're not going to have to do that for this course, so don't worry about that, okay? But you should be a little bit familiar with the muscles that are being talked about on page 103. Okay, now our goal in this chapter, in chapter 6, is not to list out all the different skeletal muscles of the body or anything like that, but to give you a basic idea of how muscles actually contract. What is the physiology of muscle contraction? But before I can cover that with you, we've got to get a little bit of basic understanding of how muscle tissue is organized in the body, okay? So for that, I'm going to turn your attention to page 104 in your textbook, figure 6.3. So we're going to go over a basic overview of muscle tissue organization. Okay, and the goal of this is to familiarize yourself with basic muscle tissue anatomy so you can understand how muscles contract. All right. Okay, so first off, so what I'm going to do is take us on a little journey, a little tour, where we start at the level of an entire muscle, like your arm muscle. Think about your biceps brachii, right? Okay, if you really don't know what that looks like, go ahead and look at page 103, and you see figure 6.2, you see the biceps brachii. So we're going to start with an entire muscle, like the biceps brachii, and we're going to progressively go on a journey together where we go smaller and smaller to examine what that muscle's actually made out of, how the anatomy of that muscle is structurally organized. Okay, so I'm going to start large with the entire muscle and get progressively smaller. Okay, so we're going to start large and go small. So number one would be an entire muscle, like the biceps brachii. Okay, and again, I'll refer you to page 103, figure 6.2, if you don't know what the biceps brachii is. Okay, and I'm also going to take you on a journey where we need to know the content that you see on page 104 and page 105 of your book. Okay, so all right, so let's continue. You know, hopefully you know what I'm talking about. Let me stop sharing for just a moment. Folks, do you know what I mean by these wooden dolls you can sometimes buy at the flea markets or at a fair, where you have a wooden doll and you open the wooden doll and you look in and there's a smaller wooden doll. And then you open the cap on that and look inside and there's an even smaller wooden doll. I think those have a certain name, but I know them as nesting dolls. Okay, do you know what I'm talking about? Okay, that's the kind of journey we're going to go on. So the biceps brachii is the largest wooden doll. I'm going to open that biceps brachii up and look inside and look at what we find next. Okay, so back to our Word document. Okay, so if we crack open our biceps brachii like a doll, like a nesting doll, and look inside, you're going to find that the biceps brachii is made of bundles of muscle cells. Okay, the biceps brachii is made of bundles of muscle cells. And let's see here. Folks, can you tell me what a bundle of muscle cells is called? Can you type that into the chat window? What's a bundle of muscle cells called? Okay, Alyssa's the first to respond and she got it. Emily, good job. That's exactly right. Okay, I'm not showing you anything that's not accessible from your own textbook, right? Bundles of muscle cells are called fascicles. Okay, so make sure you're putting your eyeball on these things. This terminology is on figure 6.3, picture A at the top. There's these cartoon bubbles, and one of the middle cartoon bubbles says a bundle of muscle cells is called a fascicle. Okay, so we have the entire muscle, biceps brachii. We open it up, we look inside to see what it's made of. We find these groups or bundles of muscle cells called fascicles. So if we were to take the fascicles as a nesting doll and open that fascicle up and look inside, what are we going to find? Well, what we're going to find obviously is individual muscle cells. Okay, now I'm going to come down here a little bit below our flow chart and tell you that an individual muscle cell is also known as a muscle fiber. Okay, so if you hear the phrase muscle fiber, that's the same thing as a muscle cell. When you hear the word fiber in biology, particularly in the context of this course, fiber basically means a chain of something or a long thread-like structure of something. Well, muscle cells typically have elongated thread-like appearances to them. They're not a typical cell that you thought of when you looked at the anatomy of a cell in chapter 3. Okay, so muscle cells kind of look like long threads. So an individual muscle cell is also known as a muscle fiber, and it is also known as a, oh goodness, I'm drawing a blank all of a sudden. It's also known as a myocyte. Okay, myocytes are muscle cells, and that's the same thing as a muscle fiber. So if you see muscle cell, muscle fiber, or myocyte, it's all referring to the same thing. Okay, so let's go back to our flow chart here. So if we opened up the fascicle, we see an individual muscle cell. Then, if we take that individual cell like a nesting doll and open it up, crack it open and look inside, what are we going to find? Well, folks, you're going to find all the components of an average cell that we learned about in chapter 3, right? You're going to find the nucleus, you're going to find mitochondria, endoplasmic reticulum, lysosomes, cytoplasm, peroxisomes, vacuoles, cytoplasm, plasma membrane, all this stuff, right? All that stuff is present. So what I'm going to focus on next is not the stuff of an average cell, which is present and which you know about, but I'm going to talk about some anatomy which is unique to muscle cells, and it's important for you to appreciate so that you can understand the physiology of how muscles contract. Okay, so an individual muscle cell, we crack it open, we look inside. The next thing that you need to know about for this chapter are things called myofibrils. Okay, myofibrils are the contractile organelles of a cell, of a muscle cell. Okay. Okay. You know from your study of chapter 3 that the nucleus is an organelle, mitochondria is an organelle, endoplasmic reticulum is an organelle, right? You know these structures are basically things that we generically refer to as organelles. Well, there are unique organelles inside skeletal muscle cells, well, in all muscle cells, okay, that are called myofibrils. So let's see here. You can also see a myofibril with a cartoon bubble on figure 6.3, picture A at the top left corner, page 104. There's a cartoon bubble there pointing to a cylindrical tube called a myofibril. Okay. I'm going to highlight the fact that the myofibrils are considered the contractile organelles of the muscle cells. Okay. All right, gang. Are you okay right now? Is everybody okay?
What page is that myofibril thing on?
The cartoon bubble pointing to the myofibril is page 104.
Thank you.
Okay. Okay. Now, if we were to take the myofibril and break it down a little bit, open up the nesting doll and kind of look at, well, what's the myofibril made of? We would find that the myofibril is made of repeating segments of anatomy that are called sarcomeres. Okay, so number 5 here are sarcomeres. Okay, sarcomeres are segments of myofibrils. All right. So if you glance over on page 105, figure 6.4, you will see at the top this cylindrical tube, and there's a bracket there that says 1 sarcomere. Okay, if we take that 1 sarcomere and link it to another sarcomere and another sarcomere and another sarcomere, we would make a chain of sarcomeres, which together is going to form the myofibril. Okay, so sarcomeres are segments of myofibrils. All right. Now, it's important that you appreciate some terminology here, right? Every field of study that you will ever encounter in life, you know this, always has its specialized terminology, right? Economists have their terminology, engineers have their terminology, business people have their terminology. Well, biologists have their terminology. Okay, sarcomeres are segments of myofibrils, and the sarcomeres are referred to as contractile units. Okay, these are called contractile units. Okay, I'm gonna underline that for you in your notes. Please take care that you don't confuse the fact that myofibrils are contractile organelles, but sarcomeres are contractile units. They mean different things. They refer to different things, okay? Please don't tell me that a sarcomere is a contractile organelle. That would be incorrect. Please don't tell me that a myofibril is a contractile unit, because that would be incorrect. So be very careful as you study. All right, so the myofibrils are made of sarcomeres, and you see an example of 1 sarcomere on figure 6.4, page 105. Any questions so far? Okay, so we're almost done with our nesting doll analogy here, but I'm going to take a sarcomere. I'm going to open the lid, so to speak. I'm going to crack it open, look inside, and see what there is to see. Okay. Well, the next thing in our flow chart here is going to be number 6. These are the myofilaments. Okay. So be very careful, because what I see, what students have done in the past is confuse myofilament with myofibril. Okay, myofibrils are the contractile organelles. The myofilaments are chains of protein that overlap each other to build a sarcomere. Okay, these are chains of protein that overlap to build the sarcomere. Okay. Now, from your reading in the textbook, or what you already know, there's basically 2 major types of myofilaments that your book's telling you about. What are the names of these myofilaments? Can you tell me that in the chat window? Alyssa, I see your question. Okay. So these chains of myofilaments, they do overlap each other, and they're shown to you on pages 104 and 105. What are the names of these things? Remember, they're going to be part of a sarcomere. They're going to be a component of a sarcomere. Okay, Catherine, nicely done. Okay, Emily, good, right? They are the actin and myosin. Very good, okay? So in your textbook, what color is actin? Okay, actin is green, right? Actin is green, and obviously myosin is purple. Very good. Now I know you folks are looking at the right thing, and that's what I need to know, okay? So these myofilaments are chains of protein that overlap to build the sarcomere, Okay, there's two types. Two types, actin and myosin. Okay, so please be aware that actin and myosin are proteins. They're proteins. And when you link one protein to another protein to another protein, you build the filament. So the actin and myosin myofilaments are chains of actin and myosin proteins that are linked together like the links of a chain at the back of a tow truck. Okay, are proteins that are linked together repeatedly to build the myofilament. Okay. All right, gang. Very nicely done. I think we're in good shape here. Do you have any questions right now? All right. The myofilaments, which hopefully you now appreciate what those things are and kind of what they look like. Those things, the myofilaments, are the business end of muscle physiology. They are where the rubber meets the road. They are where the buck stops. And what I mean by that is it's the mechanical motion of the actin and myosin myofilaments that actually empowers muscle contraction or enables muscle contraction. Okay? The further overlap the actin and myosin have, the more contracted the muscle is. The less overlap the actin and myosin have, the more relaxed the muscle is. Okay? So we're going to have to walk through this, but I think before we walk through the muscle contraction process, I'm going to show you a cartoon about this, which is going to strengthen your knowledge of muscle anatomy and introduce you to the idea of muscle contraction. Okay, so let's see here. The next major talking point for us is going to be muscle contraction. When you ask, can you strengthen the overlap? To some degree, yes, right? You know, think about it. I mean, you can have a very light contraction of your arm muscles at this time, and then you can tell your brain to contract more firmly. When you do that, when your brain tells your arm muscles to contract stronger, you are producing more overlap. Okay? All right, so muscle contraction is what we're after here. Muscle contraction physiology is largely explained by a mathematical idea, a mathematical theory, not a mathematical idea, a biological theory called the sliding filament model. Which is introduced to you on page 104. Okay, and we're going to spend some time thinking about this. But I do believe that this is one of those scenarios where probably seeing a cartoon is going to benefit you rather than have me talk to you about it for 30 minutes. So let me start by showing you a cartoon, and then we will summarize what we're seeing on this video. Okay, so bear with me. I'm going to turn to a YouTube video on this topic. Okay, so this video, which I will send you a link to, is called Muscle Contraction Process High Definition Animation. And here we go.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
Okay, folks, so sorry about that intrusion of that last video. For some reason, YouTube does that when they have free content on there. But at any rate, you could see most of the video unobstructed. So we're going to have to go through this. We're going to have to go through this in some detail. And before I get to it, do you have any questions you'd like to ask? Do you have any questions that came up from simply watching the video? Quinn, you ask, is the neurojunction unique? Well, in what aspect do you define unique? I mean, I don't find a neuromuscular junction between a neuron and an epithelial cell. I find it between a neuron and a muscle cell. So from that aspect, it's unique. Okay. Any other questions? Lasha, do you have a question?
No, I must have hit the mute button by accident.
Okay. Okay. Quinn, how much ATP does one muscle contraction take? It depends how forcefully you want to contract. The greater the strength of the contraction, the more ATP is going to have to be used because you're going to have to engage more sarcomeres, which means more myofilaments. Okay, so I would say for a light contraction, very light contraction, you know, lifting a paper clip or something like that, you're probably looking at several thousand. For massive muscle contraction where you're really straining yourself in the gym to do some weightlifting, we could be talking easily about millions of ATP molecules. Okay, anything else? All right, so as involved as that video is, it is understandable if we simply break it down. So I'm going to start breaking this down for you so we can understand it together. Okay, so I'm going to go back to sharing my screen. Okay, and I will say that I'm a little disappointed in your textbook that the authors of the well, either the authors or the publishers of the book chose to take a really complete picture or diagram of this process out and simply describe it in words. I think that's a negative aspect. They should have kept it in. And so what I will try to do later today is send you information on this picture here, which I hope you're seeing on the screen right now, which is the cross-bridge cycle. And so this image here that I'm showing you currently on the screen is going to give you a graphical, pictorial, cartoon kind of way of thinking about the things that we're going to put in your notes, okay? So I'm going to try to send that to you later today. Okay. All right, gang. So when it comes to the sliding filament model, please understand when you hear the phrase model, M-O-D-E-L, when you hear the word model in science, it means our current best understanding. It doesn't mean that it's completely correct. It doesn't mean that it's completely comprehensive. What we're saying is that here's an idea that science currently accepts as being plausible to explain some process. So here we have a plausible explanation of muscle contraction with current known scientific facts, okay? All right, so the first thing I'm going to put down, I'm going to be talking primarily about the language in your book on page 104 on the right-hand column of text, okay? You see there's bold-faced terms. And you notice that the first thing it says in the bottom right corner there is resting sarcomere. So I'm going to put I'm going to put down step 0 as the resting sarcomere, okay? Step 0 is the resting sarcomere. And what we need to say at this point is that when the muscle is in a resting sarcomere state, you know, the muscle is relaxed. Okay, this is when the muscle is relaxed. There's not any heavy contracting, no heavy lifting or force generation being created. Okay. Okay. And so in this circumstance, when the muscle is relaxed, what that means is that the myosin myofilament is not touching the actin myofilament. Okay. Muscle myosin as a protein here. Okay, if you look on page 105, figure 6.5, once again, we have the green actin, we have the purple myosin. You notice in the resting sarcomere picture that the myosin head is not physically attached to, it is not physically touching the actin protein. Myosin is not touching the actin protein at this time. Okay. Basically, at this moment in time, the muscle is relaxed and it's simply waiting on some kind of stimulus, some kind of electrical stimulus from the nervous system to begin the process of contraction. Okay. So the muscle is waiting to receive an electrical stimulus from the nervous system. I will point out that there are two things that are attached to the purple myosin head. There are two things attached to the purple myosin head. Can any of you tell me what those two things are? Matt, you asked, does this state 0 ever truly occur in the body? Yes, it does occur. You know, think about yourself at rest when you're asleep. Yes, your muscles are rhythmically contracting occasionally to help your blood circulate, but there certainly are moments of time where your muscles are about as flaccid as they can possibly be. Okay, so other questions? Okay, thank you, Catherine. Okay, so my question to you is, what are those two things that are attached to the myosin head? They're not actin filaments. So let me ask you a separate question. When you look at figure 6.5 on page 105, what color are the two things that are connected to the myosin head? What color are they? Blue and yellow. Thank you, Alyssa. Okay, now I know that we're looking at the right stuff. Blue and yellow, tiny little circles, tiny little balls that are kind of embedded there in the purple myosin head. Well, what is the blue and yellow ball? What are those things? So once again, what are the blue and yellow ball that is connected to the myosin head on page 105? What's the identity of the blue and yellow ball? Catherine, it's not an actin molecule, no. The answer is in the text of the resting sarcomere paragraph on page 104. Quinn, thank you. ADP and phosphate. Okay, Matt, I hear you. Okay. Okay, so that's exactly right. Okay, please take notice that ADP and inorganic phosphate, which is typically abbreviated PI, are attached to myosin. Okay. So what is AD? What is AD as in David? What is ADP short for? So the question is in the chat window. Emily, you're very close, but not 100%. Okay, so what's the whole thing, folks? Thank you very much, Nick. Adenosine diphosphate, okay? Adenosine diphosphate is the little brother of ATP, adenosine triphosphate, right? ADP is very similar to ATP. It's simply lacking one of the phosphate groups, okay? Well, that phosphate group has been ripped off and is present as an inorganic phosphate on the surface of the myosin protein, okay? So I have another question for you, and this one is a little bit philosophical, but I'm going to try to tease it out of you. Okay, so the question is this: where did the muscle cell get the ADP from? Where did the muscle cell get the ADP from? Okay, Quinn, you say from ATP by taking off a phosphate. I like it. You are correct. You are absolutely technically correct, but that's not the philosophical answer I'm looking for. You are correct, but, okay, so Alyssa, you say indirectly from food, so I suspect that you're thinking about aerobic cellular respiration, and that is correct also, and I love that you answered it that way. That's awesome. That means you're learning. But like so many things, there's another aspect to thinking about things, and nobody's touching on it yet. So yes, ADP is created when phosphate is ripped off adenosine triphosphate. And of course, all of these molecules are manufactured during aerobic cellular respiration, and the majority of which you know is made in the mitochondria. Those are all technically correct facts that you are correct. But we're in a scenario here where you can answer a question technically correct and still be missing the point of the conversation. Okay, gang. So I do this every semester to traumatize you on purpose, okay? Because you need to think differently here, folks, okay? In this case, the answer that I'm fishing for is that ATP was created during a previous muscle contraction cycle, okay? The sliding filament model is also known as, AKA the sliding filament cycle. Going to put that in bold, okay? We're talking about a process that is cyclical. So there's really no start, there's really no end. Defining a start is very arbitrary. It's like looking at a circle. If you look at a geometric circle, is there really a start? Is there really an end? No, there isn't. I mean, we could philosophically make it any location starting and ending, okay? But my point here is that when there is phosphate attached to myosin at the resting state, that phosphate that's there was put there at the end of a previous muscle contraction cycle, okay? The ADP and PI are created at the end of a previous muscle contraction cycle, okay? Folks, is this making sense? Is this making sense what I'm trying to say to you? Okay, does anybody need me to try to explain this differently? Are you okay? You know, going over the notes together here as we're doing and having you watch the video again on your own time, I think it's going to be really critical for you to truly get what we're talking about here. Okay. Okay. So let's keep going here and see how you do with this. Okay. So step 0 is the resting sarcomere, and it has these features that I've tried to list out in your notes. Okay. The ADP and PI that are attached to the myosin head got there by something that happened at the end of the previous muscle contraction cycle. Okay. All right. Moving on here. Step 1. Step 1. Step 1 is the cross bridge attachment. Okay. And so you do have a picture of that. Your book says cross bridge formation. That's going to be the same thing on figure 6.5, page 105. Okay. And so, you know, this will go better if you participate. This will go quicker if you participate and answer questions. So my next question to you is, you know, what two proteins are touching each other in figure 6.5 that were not touching each other during the resting sarcomere state? What two proteins during the cross bridge formation are now touching each other? Okay, good. Thank you, folks. You got it. Myosin and actin. Okay. Alyssa, you answered troponin and calcium. You're not wrong that they're touching each other, but calcium's not a protein, right? It's not a protein, it's an ion. Okay. So hold on to that, Alyssa, for a little bit down the road with some conversation that we might not get to this afternoon, but have to do it next time. Okay? All right, Alyssa, that's okay. So actin and myosin are touching each other. And the point of it is, during step 1, the nervous system has generated a stimulus to trigger the muscle contraction process to begin, to take the muscle from its resting state, step 0, into a state of actively trying to contract. Okay, so the nervous system has stimulated the muscle cell, and myosin, in response to that stimulation, myosin has literally lifted up. It has physically moved its position in the interior of the cell. It has physically moved like a nano machine, and it has locked on to the surface of an actin protein. Okay, so nervous system has stimulated a muscle cell, and myosin has moved up to attach to actin. When myosin is physically attached to actin, this is called a cross bridge. Okay, this is called a cross bridge. Now, okay. Now, obviously that myosin head cannot move without an energy source, right? Molecules don't just move randomly around without some sort of energy. And the reality of it is that in step 0, where you have the ADP and PI, well, when the phosphate is ripped off the ATP, that releases a little bit of biochemical energy that can be used to reposition the myosin head up to grab the actin, okay? So myosin is energized to move from energy released by the breakdown of ATP into ADP plus PI, at step 0, okay? All right, so the breakdown of ATP releases a little bit of energy that myosin is able to use to move itself up to the actin, okay? All right, so that's cross bridge formation, okay? Any questions at this moment? Okay, step 2. Step 2 is the power stroke. Okay, your book says bending of the myosin head, parentheses, the power stroke. Okay. All right, so in this one, here's where I really, really wish that we had a better visualization. So let me just poke up that image for you here a little bit. Okay, so notice that step 1, here where my cursor is the cross bridge formation. Step 2 is the power stroke. Okay, so take note that the position of the myosin head, which is red in this picture, it's not purple, but it's the same thing. The position of the myosin head is different in picture 2 than it is to picture 1, right? The position is different. So going back to the Word document, okay, I'm going to give you a chance to stare at this picture. Take a look at number 2 and compare it to number 1, and look at the position of the myosin head. Look how it has changed between 1 and 2. Between 1 and 2. So, okay, now I'm going to ask you, okay, in what direction is the myosin head moving during the power stroke? In what direction is the myosin head moving? Okay, thank you, Jennifer. Okay, let me bring it back up again, okay? All right, is it really moving to the right? Okay, Matt, you say to the left. Alyssa says to the left. Okay, all right, folks. Okay, you say, Quinn, it's moving left and pulling the actin left. Okay, Jennifer, that's right. Your left is the other one, right? Okay, all right. So, folks, this would work better if we were in person because it's just better. But if you have your video camera on the count of 3, I would like you to point to the left and keep your arm up to the left. 1, 2, 3. Point to the left. Okay. If we were in a room together and I was looking at you and you were looking at me, do you get that our arms would be pointing to opposite sides of the room? Do you see that? It's like a Three Stooges moment, a Laurel and Hardy moment, an Abbott and Costello moment. Do you see that left is relative to your viewing perspective? Okay, it is relative to your viewing perspective. So I don't want you to think that the myosin head always moves to the left. What I want you to think is that the myosin head is moving towards the center of the sarcomere. It's moving towards the center of the sarcomere. Okay, so let's put that down in your notes. Okay, at step 2, the myosin head moves to the center. It moves towards the center, okay? It moves towards the center of the sarcomere, pulling actin along for the ride. Okay, pulling actin along for the ride. Okay, question coming through the chat window, and I know we're almost out of time. So my question to you is this. When myosin pulls on actin, what is happening to the muscle? When myosin pulls on actin, what is happening to the muscle? Okay, Quinn, that's exactly what I was hoping you would see. Good job, Matt. The muscle's contracting. That's exactly the case. When myosin moves towards the sarcomere, pulling actin along for the ride, you are shortening the length of the myofilaments, which shortens the length of the sarcomere, which shortens the length of the myofibril, which shortens the length of the muscle cell, which shortens the length of the fascicles, which shortens the length of the entire muscle itself. So this is what's happening, okay? So it's at the power stroke that the muscle is actually contracting. Okay, it's actually contracting. All right. Okay, folks, we're basically out of time here. So let me pause and ask you if you currently have questions about current content. Okay, so here's our game plan. Next class after the July 4th holiday, I will finish this topic and we will get into chapter 7, which is the nervous system. There's some really beefy topics coming up in the course that you've got to stay focused with your study time because they're pretty intense, okay? But still, take time and enjoy your holiday, okay? We're going to take a 15-minute break and be back online for our lab time together. Please try to have a pack of colored pencils or at least a couple different colored pens nearby because your folks are going to do some drawing, okay? So I will see you back online in 15 minutes, okay? Go get a drink of water and use the